When we do an IVF cycle, we're trying to mimic what's happening in the woman's body. The ovaries here release the oocytes, and then they're fertilized on day 0. And then down the fallopian tubes, they start to clean, start to grow. And then by day 5, 6, they're into the uterus. And by day 6, 7, and 8, they're implanting into the uterine lining. Have to hatch out of the zona pellucida, which is the clear active shell that surrounds each one of the embryos that keep all the cells together until the embryo is ready to move it into the uterine lining. Then once the embryo gets out of that zona pellucida, and then it can start to move its lining. So essentially all of us have hatched. We want to mimic the woman's reproductive tract as much as we can. Day 0 is considered the day of retrieval. So that would be like when the woman the release of the eggs right out of her ovary. And this is what we see right when we get the egg out of the follicle in from the ovary. So all of this mass is just cumulus, and then this dark spot in both of these squares is an oocyte. And then mature egg has this polar body that's right up at the top here at the 12 o'clock position. And that egg means that it's ready to be inseminated or ready to be fertilized by sperm. These 2 eggs at the bottom here are immature. This one here is a germinal vesicle, meaning it has a big nuclei, a big spot in it. And then this other one here is an M1, has no polar body, so it's not ready to get in sperm place with it. It just won't do anything if the sperm enters into it. It's not ready to. There are a couple methods of inseminating the eggs. We do regular insemination where we add 100,000 ml sperm to the dish and let them do their own thing. So then the sperm just can get inside those cumulus masses and make their way to the egg. And hopefully one of them will get in and cause the acrosome to get through the zona and cause the reaction and harden the zona, and no other sperm will get in. And then the other way that we do the insemination is ICSI, which stands for intracytoplasmic sperm injection. And that is where we take one sperm and directly inject it into each mature egg. As I said before, an immature egg just isn't ready to accept the sperm. If you ICSI an immature egg, it's not going to do anything. It's just not effective. And this is a little video I have of ICSI. This big holding pipette is on the side where it holds the egg in place. This little dot here is where the sperm is moving really close. Get it as close as you can to that tip. Then you force it in, suck back the sperm in the ooplasm. So you see the rush of the ooplasm. So then you know you pierce the ooplasm. As you can see, it kind of rushed in. And then you force the ooplasm and the sperm into the egg, and then it's done. One of the main reasons we do ICSI is due to male factor, meaning that the sperm count, so there's not many sperm to work with, or they have poor motility, meaning that they start with a motility less than 40%, or poor morphology, less than 5%. Morphology, 5% normal. Frozen and thawed sperm, once we get a frozen sample, then we have to thaw it. We have to do ICSI on those samples. We won't end up with enough motile sperm with the thawed sample in order to do regular insemination. Then when we do frozen and thawed eggs, we want to make sure to do ICSI with those because the zona of the eggs can be harder. So then we want to ensure that those eggs can allow the sperm to get through. So sometimes we don't think that the sperm will get through because that's only gets hard if the eggs have been frozen and then thawed. So after 18 hours after the inseminating, and this particular one is after doing ICSI, then we look for the signs of fertilization. It's 2 pronuclei and 2 polar bodies. Obviously then the polar bodies and the pronuclei are one from the male and one from the female. The pronuclei is the chromosome packages. So you can see in this picture of the egg that there's 2 nice looking pronuclei right in the middle of it. This one on the bottom is an abnormal one. Either the egg kicked out an extra set of chromosomes or maybe 2 sperm right in there, but you really don't know. More than likely. Again, this is our slide that shows the embryo culture. Day 0 was that egg retrieval. Day 1 is our fertilization check to see how many have that fertilized correctly. Day 2 is when we see anywhere from 2 to 4 cells. Day 3, we want to see those nice 8 cells. And 8 cell on day 3 is what we would give an A. So this one is for day 3 transfer. This is a perfect looking 8 cell. All the cells are nice and symmetrical. But on day 4, we expect the embryos to be at the morula stage of development. The morula technically means raspberry. So they kind of look like a raspberry. They kind of are the cells are all smushed together, and they're anywhere from 16 to 25 cells. And then on day 5 and day 6, we want to see those blastocysts. For an embryo transfer on day 5, we want to see the blastocyst. This outer layer is called the trophectoderm layer, and this little ball right here is the inner cell. The blastocyst on day 5, day 6. About 50% of embryos reach that blastocyst stage when we go through the IVF process. The outer layer, called that trophectoderm layer, is cobblestone group of cells that will develop into the placenta, where that inner cell mass, this ball right here, will develop into the fetus, the embryonic tissue. You want a nice cobblestone layer of cells, and then you want a nice tight ball of cells from the inner cell mass. So this one we would grade a 4AA. The stage of the blastocyst is what we give the 1 through 6. A 1 and a 2 is graded as called an early blastocyst. A 3 would be called a blastocyst. 4, as this one in the picture is called an expanded blastocyst because that zona, that outer protective shell, is starting to thin. A 5 would be one that is starting to hatch out of the zona, and a 6 is one that is completely hatched out of the zona. And then we grade the inner cell mass and the trophectoderm layer A, B, and C. A obviously is good, B being fair, and C is being fair. There's some pictures that I have of good and poor embryos, and then more average looking embryos. The good embryos, you can see, have those nice cobblestone, lots of trophectoderm layers, cells around them for the trophectoderm, and their inner cell mass is a nice big ball of cells. The average looking embryos, they kind of have some thinner trophectoderm layers, and the inner cell mass is kind of visible in most spots. This one might be 20. And then the poor embryos, the trophectoderm layers all thin. And then the inner cell mass is kind of sparse. This one right here is all thin, and then these are just a couple inner cell mass cells. Not very good. You do not freeze poor embryos or biopsy those. It's just not worth the patient's time and energy. Those embryos that we knew wouldn't survive the freezing or the thaw process. So thank you for your time.